It's my pleasure today to share some of my PhD research on odor cues of MHC genotype in songbirds. This research was completed at Western University in London, Ontario, Canada. We can all probably imagine what we find attractive in a potential mate. However, it isn't necessarily just physical characteristics that we respond to when we think about mate attraction. And a perfect example of this comes from studies performed on the role of smell in human mate choice. Female undergraduate students significantly preferred the body odor of male undergraduate students when they were genetically different from the females at an important immune gene region called the major histocompatibility complex. MHC is a gene family that encodes molecules that recognize and bind to pathogen-derived antigens. MHC is both polygenic, having multiple genes, and polymorphic, having multiple gene variants or alleles. There are two main classes of MHC. Class 1 is involved in defense against intracellular pathogens, while Class 2, the focus of my talk today, is primarily involved in defense against extracellular pathogens such as bacteria. MHC-based mate choice can confer both direct and indirect benefits. Under direct benefits models, individuals should choose a mate whose genotype at MHC is diverse. These more heterozygous mates will be better able to defend against a wider variety of pathogens, meaning they should be healthier and better able to provide high-quality parental care, territory, or both. Under indirect benefits models, or compatible genes models, individuals should choose a mate whose genotype at MHC is dissimilar from their own, and this serves to increase the genetic diversity and, presumably, fitness of the resulting offspring. MHC-based mate choice is indeed common across taxa. In addition to the human example I gave earlier, fish also appear to make decisions based on the MHC genotype of their potential mates. In this study on three-spined stickleback, female fish were given the opportunity to spend time with odor from either a similar or dissimilar male at MHC class 2B. Females spent significantly more time with the odor from the dissimilar male, suggesting they prefer this odor, consistent with predictions of compatible genes models of mate choice. In birds, olfaction has historically been understudied, largely due to the myth that birds had little to no sense of smell. All birds have a fully functional olfactory system, and they use smell in a variety of contexts, including in their social and mate choice behaviors. I use preen oil as a proxy for body odor in birds. Preen oil is secreted from the uropygial gland, which is a large organ located at the base of the tail in most bird species. Preen oil is complex, containing up to hundreds of compounds, and it has multiple functions. Classical functions include things like waterproofing and feather maintenance, but more recently, we've been learning that preen oil chemicals contain information that may be important for the birds. For example, in some of my PhD research, I found that there are significant differences in the chemical composition of preen oil between the sexes in song sparrows. To summarize this introduction, MHC-based mate choice is common across vertebrate taxa. Benefits of MHC-based mate choice may be tangible, direct benefits, or intangible, indirect benefits. Odor cues are involved in MHC-based mate choice in taxa including fish and mammals, and it's possible that birds might attend to odor cues in preen oil in a similar fashion. To explore this further, I asked whether preen oil provides odor cues of MHC genotype. I hypothesized that songbirds can use preen oil odor cues to assess the MHC genotype of potential mates. And I predicted that songbirds would spend more time with preen oil from MHC-dissimilar and MHC-diverse potential mates. I used a two-choice design to test this. Birds were placed in a Y maze and given 10 minutes to spend time with preen oil collected from either an MHC-similar opposite-sex conspecific or an MHC-dissimilar opposite-sex conspecific. Then, I recorded the amount of time they spent with each odor sample. On the y-axis, I have the amount of time spent with a given sample, preen oil from an MHC-similar or dissimilar potential mate in panel A, preen oil from a more or less diverse individual at MHC in panel B. In the first case, I found that song sparrows spent significantly more time with preen oil from an MHC-dissimilar opposite-sex conspecific. compared to the time they spent with an MHC-similar one. That's shown by the large open and filled circles in the plot, and the small gray circles joined by a line show the individual responses of each bird in the test. Similarly, I found that song sparrows spent more time with the odor of an MHC-diverse individual. That's an individual with more MHC class II alleles compared to the other individual. I concluded that songbirds can use prenoil odor cues to assess MHC genotype of their potential mates, and their preferences are consistent with compatible genes models of mate choice, preference for an MHC-dissimilar possible mate, and direct benefits models of mate choice, preference for a more diverse potential mate. MHC genotype might affect body odor in several ways. MHC molecules or bound metabolites could be odorous, or MHC molecules might carry volatile odorants. These could then be excreted from the body directly through the urine, sweat, or prenoil, providing a direct effect of MHC genotype on body odor. Because MHC class II is involved in extracellular pathogen defense, for example against bacteria, it's also possible that MHC genotype could shape the microbiota that are able to successfully colonize a host, and this could indirectly affect body odor. In terms of the relationship between MHC genotype and prenoil, if there's a direct effect of MHC on prenoil, I would predict there to be a strong correlation between MHC genotype and the chemical composition of prenoil. If there's an indirect effect of MHC on the composition of prenoil, I would predict that there be a strong relationship between MHC genotype and the Preen gland microbiota, a strong relationship between Preen gland microbiota and prenoil, indicating this indirect effect of MHC genotype on the composition of prenoil that's mediated through microbes. To explore these indirect and direct mechanisms, I collected prenoil, blood, and Preen gland bacterial swabs from a number of song sparrows. I chemically analyzed the prenoil and used next-generation sequencing to genotype the birds at MHC, as well as to characterize the Preen gland microbial community. I found that MHC genotype was significantly correlated with prenoil, such that the more similar birds were at MHC class II, the more similar their prenoil chemistry was. Similarly, the more similar birds were at MHC class II, the more similar their Preen gland microbiota were. However, there was no relationship between Preen gland microbiota and prenoil chemicals. This suggested that there was a direct effect of MHC genotype on both prenoil and Preen gland microbiota, but that there was no relationship between the microbiota and the prenoil composition. As I mentioned at the start of this talk, though, prenoil is a complex mixture of hundreds of compounds, and it has multiple functions. And this is also true for microbiota. So I ran a post-hoc analysis to look at whether any subsets of Preen gland microbiota could potentially be driving a relationship with prenoil chemistry that was masked by using the full and more complicated data set. When I did this, I found that indeed a subset of Preen gland microbiota was significantly correlated with prenoil chemicals. This suggested that it is possible that MHC genotype also affects prenoil composition indirectly through an effect on the Preen gland microbiota. To summarize, songbirds use prenoil odor cues to assess the MHC genotype of potential mates. MHC genotype predicts both the chemical composition of prenoil and the Preen gland microbiota, and subsets of microbiota of the Preen gland predict prenoil chemical composition. Prenoil odor cues are a likely mechanism for MHC-based mate assessment in birds. I showed for the first time in a passerine songbird that prenoil odor cues are used in MHC-based mate assessment, and birds' preferences aligned with both compatible genes and direct benefits models of mate choice. Song sparrows of both sexes showed preference for both MHC-dissimilar and MHC-diverse opposite-sex conspecifics. MHC genotype might influence host odor both directly through an effect on the composition of prenoil and indirectly through an effect on the Preen gland microbial communities. To better understand the relationship between MHC genotype and body odor, an important next step would be to experimentally alter or disrupt the Preen gland microbial community and test whether this affects birds' ability to discriminate MHC genotype using odor cues in prenoil. To conclude, I'd like to thank my PhD advisor, Dr. Beth MacDougall-Shackleton, my collaborators, Drs. Mark Bernards and Greg Gloor, many lab members and other faculty at Western who've supported my research over the years, as well as my funding sources, research stations, and of course my study species, the song sparrow. Thank you.